My name is Korets-Smith. I'm Chief Business Officer and co-founder of Virica Biotech. It's my pleasure to tell you a bit about the company. I think we can all agree that gene therapies and cell therapies, really viral medicines in general, are going to play a huge part in human healthcare. I think we can also agree that these therapies have a very common challenge, and that is manufacturing. Because of the kind of industry this is, manufacturing really is the product. And we also know that it's complex, it's expensive, and there's just not enough capacity in the world to manufacture for all of the clinical trials, not to mention for all the approved products that are eventually going to be coming to the market. In some cases, manufacturing can really become a limiting factor for what we can do with our gene therapies. Now, what we and others know and is becoming more and more in the public eye is the fact that one of the main reasons for challenges in manufacturing of viruses, viral vectors, are the antiviral defenses that are present in all host cells. Now, these antiviral defenses are very complex and are triggered in different ways in different cell lines by different viruses. But we absolutely know that they're there, and they are, in fact, reducing the efficiency of manufacturing. This is something that is typically, I would say, has been ignored in this particular space, though of course is very well known in other areas, like, for example, oncolytic viruses. But what Virica is doing is bringing this particular issue to the forefront and presenting a solution to the problem. The company was founded on the basis of technology developed by the Ottawa Hospital Research Institute, and our co-founder, Dr. Jean-Simon Diallo, who actually was originally interested in oncolytic virus biology. He was looking at how to make oncolytic viruses work better. And through his research and through thinking about it, he actually did what we believe to be the world's first high-throughput pro-viral screen. Effectively, he was looking for molecules that make oncolytic viruses grow better. And he found them. He found an entire library of them. We have over 130 under patent now. But then he said, wait a minute. If I can make oncolytic viruses grow better, what other viruses can I make grow better? What other applications can I have for this technology? And that is how the viral sensitizer platform was born. The platform itself, as I mentioned, is a collection of small molecules that fundamentally do one common thing. They reduce the antiviral defenses that host cells have to incoming viruses, thereby allowing the virus to come into the cell and do its job, whether that job is replication, transduction, whatever it might be. And so because we have this library, we can now achieve two different things, and we can offer our clients solutions to two related problems. One is to offer a more efficient manufacturing process through the addition of these small molecules to the upstream of viral manufacturing. And secondly, we can offer an improvement in transduction efficiency, meaning more efficient production of therapeutic cells like CAR-Ts and KCARs. Now, we do this not with 130 compounds, because that's a little much. We start with 8. These are what we call our workhorse compounds, and they are a cross-section of the chemical entities that exist within our library. With these 8 compounds, we begin formulating specific combinations for our clients' manufacturing platforms. And we've done this for the past 10 years. The technology exists. We've demonstrated that these molecules can be effective at improving yields significantly with a variety of host cell lines and a variety of virus types. And we have data for a lot of these now, and we are generating more data with our clients on a variety of other virus types. Of course, the major interest in this room is lenti and AAV, and I can tell you, and I'll share some examples of how we improve the efficiency of manufacturing for those viruses. The incorporation of the technology is quite simple. It goes right into the upstream process, and then the process goes on as usual. So we can work with any technology, any manufacturing substrate. at the end of the day, all we have to show is that these are gone at the end of the process, and we have analytical methods to do that. Let me share some examples. This is AAV. This is actually third-party data generated by our clients. There are 8 molecules here, and what you can see is with just a single compound, we can improve the efficiency or the viral output 2 to 3x with 6 out of the 8 compounds. Now imagine if you can actually combine those together, what will happen? Well, I'll tell you, in the lentivirus example, this is what we've done in-house. We've done just that. We did single compound formulation and then double compound formulation. And what you can see is that we can improve the viral output in small scale, 96-well plate, 7x, and 5x when you scale up to T75 or T175. So this scales very, very well, and we know the combination of compounds work better than single compounds. We can also work with other intensification platforms, like, for example, the Acellus here. So this is an example with MVA, where we can improve the yield in small scale 10x with 9 genetically different types of MVA. And when that's transferred into an Acellus, you still get a very nice 4x improvement in yield. So it can be very compatible with other technologies, not just Acellus, just about any other. And here is some preliminary data, but indicative, to show our ability to improve transduction efficiency. This was an experiment done with 3 different virus types, and what you can see is that we can enhance the transgene expression from AD, Lenti, and AAV in a very significant way. Again, this demonstrating that this could be very interesting when looking at therapeutic cell production. I think it's important to note the regulatory path that this technology will be following is one for a very common contact material, essentially. So what we have to do and have done is develop methods to detect these compounds in our clients' cultures so that they can prove that these compounds are no longer present in their final product. We have set up the supply chain for these molecules to support our clients. It's fairly simple because these are small molecules, they're chemically defined. So we have a third party that's producing these compounds in GMP with about a 3-month lead time. All this to say, we are building up a package to support our clients as they go into the regulatory process, into the clinic, and then eventually into commercial production with their products. So how do we work with customers? That's one of the key questions that I get asked as a chief business officer, and the answer is we've listened to what our clients have told us. We are going to fundamentally enter into long-term supply and use agreements for compositions of molecules that work for your particular manufacturing platform. Before we do that, though, we have to identify which molecules work for you. And we have found, and I'm happy to answer questions outside or connect to the technical team, we have found there to be quite a bit of variation based on the cell line, based on the virus, et cetera, et cetera. So what we do as a first step in our relationship is an evaluation, which can be done either in your hands or in ours, meaning under an MTA, where we transfer 8 compounds to you for formulation, or as a service in our labs. Now, I should say we are experts at high-throughput virology. That's the way that these molecules were actually discovered originally in the first place. And so when we do the service, or if we do this evaluation as a service, we deploy that high-throughput technology. We miniaturize your process and then do the screening in as small a volume as possible in order to get the most replicates as necessary. And then we transfer back to you for scale-up and validation in your hands, which makes the most efficient process. Following either this MTA or service agreement, then we start talking about the commercial terms, the long-term supply agreement, use agreement, et cetera. And that's what we're deploying with quite a number of collaborations at the moment. I want to tell you a little bit about Virica as a company. And as I said, the last couple of weeks have been a bit of a coming out party for us. We truly believe in the promise of viral medicines. And it is our job to deliver and help you deliver on that promise, thereby expanding its use globally. We have just week and a half ago, closed a Series A round, which is very exciting for us. This allows us to actually continue to build on the momentum we've started. The round is led by Dynamic Capital, who some of you might know are a fabulous group of individuals who've been very deeply entrenched in the manufacturing space for a very long time. We're very grateful to them and happy to welcome them to the Virica family and look forward to working with them going forward to build this business. I've told you that we were founded by an individual who's very well known in the oncolytic virus space. Dr. Jean-Simon Diallo is very well published in that space and is one of the preeminent scientists in that field, as well as an entrepreneur and a biotech professional. We have a management team, including myself and our co-founder, Ken Newport, executive chair, that are complementing each other very well in terms of our skill sets. And we are very passionate about this business and about this company. The company has over 10 years of high-throughput virology experience, and we're going to be deploying that on a regular basis with our clients to support their formulation development as we grow some additional scale-up and, frankly, additional technology development as well, as this is something that we feel we're going to have to keep doing in order to stay ahead of the curve and, frankly, to support our clients. And we just recently announced that we've opened a lab facility in Ottawa in order to do that. We are very pleased to see the interest that our clients are showing in our service capacity. And so for that reason, our labs are expanding in order to do more of that and process more programs through our facilities. Our IP position is very strong. We have 5 patent families. We're building on that. And frankly, the momentum in this industry, I think, is very well received and very well represented by what we are seeing going through our labs as well. I do very much hope that we get to talk to one another. I'm here for another couple of days, but happy to connect online as well if that would be useful. Tell you a bit more about the technology, connect you with our technical team. We love talking about this stuff. And we want to talk about all the different wonderful work that you're doing, all the different programs that you're putting forward, and seeing if we can support your manufacturing processes, reduce some of those bottlenecks, and make manufacturing not a limiting factor any further. That is my contact information. Thank you so much, and happy to take questions, I believe, outside. Thank you.